Hi, let's go over what we were doing in this Find a Virus project, because I want to make sure that you get this. So it doesn't matter that we're finding viruses. We can use these tools to find anything. We just happen to be finding viruses today because they're little and they're fairly easy to work with. Compared to like a bacterial genome, which may have 1,000 genes, the viruses that we're talking about, they've got like 6, maybe 8 genes. Not bad at all. All right, so the tools we're going to use are going to be BLAST. Now, BLAST, remember, is a search engine that can search nucleotides or search proteins, and it can search nucleotides with proteins, proteins with nucleotides, because you can translate any nucleotide sequence and figure out what protein it could make. And so you can search against theoretical proteins, proteins that should exist. And so this is basically what we're searching is GenBank. It's all the sequences that humankind has recorded since the dawn of molecular biology, not time. And the version we're going to use is going to be tblastn. So tblastn lets us take a protein that we know and lets us search a database full of nucleotides. And it'll translate the nucleotides and then let us search them. And it does that all by itself. We just feed it protein and say, go look here, and it'll do that. Yeah, it's pretty nice. All right, so click it. Boom, go in there. What did you do? You put your thing into this box. You put your protein sequence into there, and remember how we found the protein sequence. We got it from ViralZone, something like that. You could have got that from anywhere. I could have given that to you. It doesn't really matter. Any protein you put in here, you can then use to search. And so they say the thing that you're pasting in is called a query. That's like the question you're asking of the database. So you paste it in there, and we changed it to this thing, the transcriptome shotgun assembly, because we want to search in transcriptomes, in RNA that was inside cells. All the genes are a genome. All the transcripts, which are messenger RNAs, are the transcriptome. That's what it means. Yeah. All the proteins, those are the proteome. Yeah, these are just the words we use when we're referring to all of a thing from inside of a cell. All the lipids are something like the lipidome. Yeah, I don't know. People don't usually use that, but there are a lot of different lipids out there. So we're going to search in this database because I know there are lots of viruses in that database. There are many other databases you can search, and depending on what you're looking for, you look in a place that might have it. A good standard place to search is going to be like the core nucleotides. That's going to have all the things that already have names, for example. We're trying to do a particular thing where we're trying to find undiscovered viruses, and the TSA is a great place for undiscovered viruses because there's a lot there. I know this because I've looked. Yeah. And then you're going to limit it by some animal group, and so in this case I put in spiders, arachnids, and that's fine. The bigger the group, the more likely you are to find a virus. So you did that, you hit blast, everything is good. Once your blast was done, you came back with a result that would look something like this. What do we have here? Well, we have a list of a bunch of names and a bunch of accession numbers. And if you look up the top, it'll say accession. This accession number is a number you can click, and here, let's click this one, and it'll take us to a page that has that sequence. So there it is. Yeah. An accession number is just like a link, basically, that'll take you to a particular gene. In this case, a gene from the TSA database, and the gene is going to be something that is actually a virus, but the people sequencing it didn't realize that. Let's interpret these. So what do you need from here? You need the length, and so that's the accession length. That's what's up here. And so this tells you how long the thing was. A full-length virus of the type we're looking for is going to be in the 8,000 to maybe 12, 13,000 range. And so these little guys, those are going to be partial viruses. We don't want to work with fragments today, just because I want you to find a nice complete thing. So you picked something that was, 8,000 or bigger, I would say. And that's great. Okay, what else is on here? The stats are useful. So the e-value is the chance of error, and if the e-value is 0, it means it's not an error. This is definitely a match of some sort for the thing you put in. So it's the same kind of sequence, not the identical sequence. And that's great. The scores are just internal things. You don't have to worry about those. The description over here, this is the name of the animal that your virus was found in. And so the first two parts here, like Pardosa pseudoannulata, if you were to Google that, let's get a new tab, Pardosa pseudoannulata, and you're going to get beautiful pictures of, oh my gosh, a spider eating a fish. Yeah, that's a Pardosa spider, Pardosa pseudoannulata. So the sequence that we're looking at is going to be a virus-like sequence from inside of that spider. Hey, cool. Yeah, probably that's a spider virus. Yeah. All right, so you understand that, and you can click any one of these accessions, and so you clicked some particular one, you went to a page that would look something like this. If you scroll down, it's got a big sequence. Now you can look at this, and you can tell this is a nucleotide sequence. How? Because it's got A, C, G, and T, and nothing else. If we're looking at a protein sequence, it's going to have most of the letters. 20 of the letters of the alphabet will be in there. But nucleotides are just A, C, G, T. If you scroll up to the top, it'll tell you whether it's RNA or DNA, and so this one's an RNA sequence. And you're saying, hey, but Dr. Newman, RNA has A, C, G, and U. Yeah, you're right. But they put it down as T, and then they just tell you at the top that, oh, it's RNA. So in your mind, you got to change it to U. Doesn't matter. All the prediction programs we're using can understand U, and they can understand T, and they don't really care which one it is, because you can have genes that come from RNA and genes that come from DNA. It's okay. All right, good. Yeah, here once again, it's going to tell you what kind of spider this came from. So mine's from a Pardosa pseudoannulata spider. Super neat, yeah. And yours may not be from a spider. Yours may be from a fish or a plant or whatever. But Google it. Figure out what it is. Learn a little biology. What you've got here are the nucleotides, and they have a bunch of numbers in between. If you try and copy that, it copies the numbers too, and the spaces, and gross, yeah. So there's a button we can click, and we can go to FASTA format. FASTA format is a neat format for storing sequences. It has two components. The first is a title line that starts with the greater than character. That's this thing, if you're new to math, yeah. The greater than character, then it's going to be followed by the accession number, and you can put that accession number in right here, NCBI nucleotide, and it will come back to this exact page. Yeah, I'm not going to do it because it'll take a second, and I'll have to reformat it. Then there's a title, and you can make the title be whatever you want. And then there's the return character, like enter, on this side of your keyboard. And then there's the sequence. And so, yeah, that's what you did. You took the sequence just like that. Once you had the sequence, you would have then translated it. So let's translate it to see what sort of proteins we can make. Because we can't really do anything with nucleotides. Nucleotides become proteins, and proteins we can understand, thankfully. So that's why we're going to do this. We are going to put it into something like Expasy Translate here, paste it all in there, translate it up, and we're looking for an improbable thing. We're looking for a very long protein, because we know that when we're looking at just junk, there will be starts, there will be stops, and it'll just look like this. It'll look like a hash, yeah. Just nothing, nothing, nothing. And then, oh, look at that. We'll have some of these big proteins. You might have one giant protein. You might have two. So there's the front half of it. And here, mine came out in 3 pieces. Weird, but viruses sometimes do that. And so I would take this piece, because it comes from the front, like the first half. And then I would take these 2 pieces, because they come from the second half. Oh, and there's the middle piece there. So first piece, second piece, third piece, fourth piece. I got a 4-piece virus. Wow. What did you get? Did you get a 1-piece virus? Yeah, that's lucky. That's convenient if you did. But yeah, these exist too. Okay, then you're going to take that, and the next thing you did was to run it through HHpred. And so you're just going to paste those in here, and bam, whoops. Those are the nucleotides. We don't want to paste those. HHpred is good at dealing with proteins, and it will tell you what your protein is similar to. Like, if there's a new kind of car, how do you recognize that it's a car? Well, it's probably got 4 wheels, it's probably got an exhaust pipe, probably has doors, probably has a steering wheel, unless it's a Cybertruck. I don't know what they have, some weird joystick device or something. But you'll be able to recognize it because it's like a general type of thing. HHpred is awesome at recognizing general types of proteins. And if you have a big protein, that's okay, because there are probably different parts inside of it that do characteristic things. We call these parts domains. It's like a part of a protein that'll fold up and work on its own. It'll like do something on its own or make a shape on its own. Yeah. So dump those in here. And there are 3 really good databases, and if you've searched these 3 and you don't find any explanation for your protein, there's probably no explanation for your that part of the protein. So those are going to be the PDB, Protein Data Bank. That's all the structures in the world. PFAM-A and NCBI conserved domains. Those are kind of farther reaching. Those are things that we may not have solved a structure for, but we've identified things that are repeating in nature, things that come up in different contexts. And then you'll hit submit, exactly like that. And you'll get a result, something like this. Let's pull this over, because we don't need to see me. Yeah, I'm fine down here in the corner. Nobody puts baby in the corner. I put myself in the corner. On the HHpred result, this, here, we can extend this whole line. This is just showing you that this is where the start of your protein is. So your protein was 2,268 amino acids long. If you didn't know that, now you do. Yeah. And so this is saying that, okay, these are different parts of your protein. If you want to measure it, you can say, all right, about 177 to about 508 is this thing. What we're looking for are columns, and you want to see how many columns of things you have. So I've got 1, 2, 3, 4, and 5. It's only one thing in that column, but we'll call it a column anyway, because it's occupying a different space here on our little line at the top. So I've got 5 things that I can probably annotate. Annotate means I can say what they do, like what they are. We got our little color scheme over here. So if we can find one of these proteins with these words, we're going to be maroon. If you find one of these, it's going to be red. Find one of these, it's going to be yellow. And if you can find something like a capsid, the first one's going to be green, the first one that appears in your sequence, and then blue, and then purple. Yeah. And we're just going to color in the sequence, and it's going to look great. Yeah. All right. So let's go back. All right. And the thing I told you to do was to wrap the sequences. So we'll do that. And you hit that once, and it just puts each one on one line. And that's fine. You just have to scroll to the end to see the end of it. All right, so pick a column. Let's pick this column. You can just mouse over it, and I'm looking for my magic words, and there's one of them, RNA polymerase. Polymerase, that's the one that we're going to turn yellow. So let's click it. Now we've gone there. It's the thing at the top, 3BSO, that's the one we clicked. Now this is the start of it, and so the second line here is always going to be your protein that you put in. And so it's saying that yours matches up with a known bona fide polymerase, and so here's the bona fide polymerase that it matches. That's not as important for you. You can look at the stats, and if you got an e-value less than 0.05, this is 10 to the minus 55th power. That's a real small number, real close to 0, so that's good. That's a chance you're wrong. The e-value is the chance of error. In this case, we are probably not wrong. So yeah, we can believe this. Yeah, we're confident. And so you find this sequence, GLPIF, whatever, you can find the whole thing in your protein that you've saved. The protein is this thing. You're going to copy this and put it in the document. Yeah, just as a place to keep it. Yeah. Because you can buy expensive programs that'll do that for you, but they're not better than Microsoft Word, weirdly, or Google Docs. So yeah, there's the start of it. Now here's the big brain thing. You got to scroll all the way down so you can scroll sideways. Yeah, and so you scroll right to the end, and there's the end of yours. And so boom. So it's saying it's position 2266 out of 2268, so that's pretty close to the end. And you can find these in your sequence, and it's still the same stuff. The second line is your protein, and the this one that's 3 from the bottom is the thing it matches. Yeah, just if you want to check the work of HHpred. So you got your HHpred annotations, you turned your things the different colors, and now you have annotated the parts of the proteins that were encoded on the genome of the thing that's probably a virus, because it has virus-like genes, and we found it. It was in a pile of RNA from inside an organism. Yeah, this is how you find new viruses, and you just did it. But more importantly, if you were given the genes from a bacteria, you would do the same thing. You take the genes, you would translate them with something like Expasy Translate, because you'd find where are the protein coding sequences, and they'd be the big pink blocks in there, same as always. Once you've got those, you'd copy them into something like a Word document. Then, for each one of those, you can run it through HHpred, and HHpred will tell you if this is a thing that belongs to a class. Is it a car? Is it a bike? Is it a bird? Whatever. Only it's going to tell you a particular kind of protein. And that's great. You will then have annotated that particular protein. And normally when you're doing this for real, you would also note down the E value and possibly also the probability. And I like that probability, 100% probability. Yeah, that's good enough for me. So there you go. That's what the project was. That's what you were doing. It doesn't matter that you were doing it with a virus because it's the same basic bioinformatics techniques. Finding nucleotides, turning them into protein, and then annotating the protein to figure out what it does. That's bioinformatics in a nutshell. Everything else is just, I don't know, a little bit of fancy rearranging the deck chairs kind of stuff. So hope that made sense. If not, you can sure re-watch it. And if not, you can sure come ask me for help because that's literally what they pay me for. All right. Thanks a lot.